Hello students, welcome to BioWise with Aanchal. I hope day 1, unit 1 is done and you could solve the questions that was given. Please children complete the entire unit to ensure that you have completed the portion, at least the part which is already done. Today we are going to start with unit 2, that is plant physiology. In this unit we are going to cover absorption by roots, transpiration, photosynthesis and chemical coordination in plants. So let us begin with absorption by roots. In this chapter, we are going to study about the transport, the 4 types of transport that you are having in your syllabus, along with it the structure of the root, hypertonic, hypotonic, isotonic solutions, fate of different cells, then different types of pressure, which is root pressure, wall pressure, osmotic pressure, and the transpirational pull, ascent of sap. Yes, so let us begin with it. Before going into the chapter, you should know how to draw the root hair diagram. To save our time, I've already drawn the diagram to show you the different layer which has to be shown in the exam. Now here is the importance of the first chapter, that is cell, where you should know that a plant cell should have a cell wall, a cell membrane, a large vacuole. So your root hair should have a vacuole, right? It's a vacuole. Then it has got the nucleus and it has got the cell membrane and the cell wall. Both of them are present. And it also shows that it's a unicellular extension to increase the surface area for absorption. Moreover, the sap is highly concentrated. So the movement of water is always from the soil into the root cell. A type of special characteristics that the root have so that it can absorb water. So the question can be, state the characteristics of the root hair which helps it to absorb water from the soil. So first you will say that unicellular extension, so that the diffusion is easy, and at the same time the concentration of the cell sap in the root is high, so that the water tends to move from the lower concentration to higher concentration. We'll be talking about hypertonic, hypotonic solution. You'll understand it better. Now, depending upon types of transport, you have 2 types of transport. One is active and one is passive. Now, active transport means the molecule has to move from a region of lower concentration to a region of higher concentration by expenditure of energy. Here the plant has to spend energy for this transport to happen. But in passive transport, you will realize that there is no expenditure of energy. The movement of molecules is always from the higher concentration to the lower concentration. Okay. Now I will give you a very good example. For example, you have a small pit or a well. If it rains, then the water has to go into the well. You don't have to spend energy. But when you have to take that water out of the well, you need to use mechanical energy or pulley to take that water out. So when the water is moving against the concentration gradient, that is from a lower potential to higher potential, you have to spend energy. But when the water has to move from higher potential to lower potential, then there is no expenditure of energy. This is what the transport is all about. Active transport, expenditure of energy. Passive transport, no expenditure of energy. There are 3 types of passive transport. One is imbibition. This happens basically with the dead cells or the dead tissues. The first water that is absorbed, adsorbed by the seed coat, so that the seed can absorb water and swell and soak. That is done by imbibition. Even in rainy season, the wooden doors tend to swell up. That is because of imbibition. Okay. Next one is diffusion. Now, if I spray a perfume at the corner of the room, slowly and steadily you will find that the entire room starts smelling or giving that fragrance of the perfume. That is because of diffusion. Diffusion is movement of the molecules from a region of higher concentration to a region of lower concentration till the concentration becomes same throughout. But osmosis. Now here, next, osmosis is very important. The first prerequisite for osmosis to happen is a semi-permeable membrane. Always remember, here also in active transport also we need semi-permeable membrane. In osmosis we need semi-permeable membrane. So here and in osmosis, it is the water molecule which moves. So the movement of water molecules from the region of its higher concentration to a region of its lower concentration through a semi-permeable membrane is called osmosis. Okay. Now, if water moves in the cell, it is endoosmosis. If water moves out of the cell, it is exoosmosis. So, let me make the things clear with the terms, that is hypotonic, hypertonic and isotonic. Yes, now we talk about the different types of solutions. There are three types of solutions. First is hypotonic, and then you have isotonic, and you have hypertonic. Okay. The dilute solutions are called hypotonic solutions. The concentrated solutions are called hypertonic solutions, and with the same concentration as that of the cell, it is called isotonic solution. Same concentration as that of a cell is called isotonic solution. Now, suppose you have got a plant cell. You know that plant cell has got a cell wall. So, I have drawn the cell wall. It has got the plasma membrane or the cell membrane, and it has got the large vacuole and the nucleus. Okay, this is your plant cell. If this plant cell is placed in hypotonic solution, hypotonic solution means it is dilute as compared to the cell sap. So, what will happen? Water will start moving into the cell. Okay, water will start moving into the cell, that is endoosmosis will occur. Okay, and the cell membrane will stick to the cell wall. Right, the cell membrane will stick to the cell wall. The cell will become fully distended, or you can say turgid. Okay, and the pressure because of this fluid on the walls, that is on the cell wall, this pressure, pink thing is pressure. Okay, this is called the turgor pressure. This is called the turgor pressure. Is that clear? At the same time, to keep the cell intact, the cell wall also exerts pressure on the inner content of the cell. This is called the wall pressure. Okay. So, a fully distended cell, the turgor pressure is equal to the wall pressure. Is this understood? The turgor pressure is equal to the wall pressure for a fully distended cell. Now, if suppose the cell is if suppose this cell is placed in a hypertonic solution. Hypertonic solution. So, what will happen? The water from the cell will start moving out. That is, the water will start moving out. Exoosmosis will occur. Exoosmosis will occur, and the plant will shrink. Since it is a plant cell, the cell wall will remain as it is, but the plasma membrane is going to shrink. Okay, so you have the cell, this is the cell wall, and the plasma membrane is going to shrink with the nucleus and a small vacuole. Okay, so when you see this place, that is between the cell wall and the cell membrane, it is filled with this solution, hypertonic solution. The cell membrane is this one, and the cell wall remains at its place, that is cell wall, cell membrane, and this place, that is between the cell wall and the cell membrane, is filled with hypertonic solution, because cell wall is freely permeable and cell membrane is semi-permeable. So, the cell wall is going to allow whatever the cell is in, the same solution will be present between the cell wall and the cell membrane. It's not water, only water. It is entire hypertonic solution. Okay, and the cell will shrink. This is called flaccid. Generally, flaccid term is used for the animal cells. For plant cells, we use the word plasmolysed. Okay, plasmolysed. So the cell with the with the cell membrane which is shrinked because of the loss of water, because of exoosmosis, is called plasmolysed cell. When you place this cell again in the hypotonic solution, the cell wall will retain its position. So this is called deplasmolysis. So plasmolysis is shrinkage of the cell membrane, and deplasmolysis is regaining its because of absorption of water when placed in hypotonic solution. So whenever a cell is placed in hypotonic solution, it gets bulged out, swells up, or you can say endoosmosis will occur and the cell will become turgid. When it is placed in hypertonic solution, the cell will shrink, plasmolysed, become flaccid. Flaccid we generally use for the animal cells. In this case, in plant cells, use the term plasmolysed. Plasmolysed cell, when it is placed in the hypotonic solution, it again regains its original shape, that is deplasmolysis will occur. Okay, and in isotonic solution, the net movement of water is zero. Net movement of water is zero. That means endoosmosis is equal to exoosmosis, so the cell will remain intact. In hypertonic solution, endoosmosis is more than exoosmosis. In case of hypertonic solution, exoosmosis is more than endoosmosis. So when you talk about net movement of water in case of isotonic is zero. This is all about tonicity. Now let us talk about osmosis little bit in detail because it's an important topic for your exams. So next, osmosis. Osmosis is the movement. If you to define osmosis, osmosis is the movement of water, is a process by which the water molecules move from a region of its higher concentration, that is dilute solution, to a region of its lower concentration, that is a concentrated solution, through a semi-permeable membrane. So you have to remember that osmosis happens only in the movement of water happens. Water molecule moves. Okay. Now here in labs and all, we can use gourd bladder, we can use cellophane paper, which acts as a semi-permeable membrane. But remember, muslin cloth is not used as a semi-permeable membrane, it's freely permeable. So muslin cloth is not used. So you can be asked questions where the experiments can be modified using muslin cloth and all, and you have to say that whether osmosis happens or does not happen. Always remember, whether it is a potato osmoscope, that is, the potato is cut into a well and then it's placed in a solution, that is sugar solution. It's a sugar solution, and here you have distilled water. So what will happen? You know that water will move out. So the level, the initial level and the final level will be different. Similarly, if you place sugar here and water there, then the initial level will be low and the final level will be high, showing endoosmosis and exoosmosis depending upon the concentration of solution it is placed. Always remember, water always moves from a dilute solution to a concentrated solution. Is that clear? So see where the dilute solution is present, and then that water will move from that region to the other region which has higher concentration. So this is all about osmosis. Now there is something called osmotic pressure. Osmotic pressure. Now osmotic pressure is the minimum pressure required to stop the process of osmosis. So osmosis is the minimum pressure required to stop the process of osmosis to happen, right? So you can have the same setup where you have the thistle funnel which is being covered with the semi-permeable membrane placed in distilled water, and here you have sugar solution. Here you have piston. So you know that from distilled water, the water will move into the sugar solution, correct? So water level will rise in the thistle funnel. So that is showing endoosmosis is happening. But the amount of pressure that you put so that the movement of water stops is called the osmotic pressure. Is that clear? This is very important when you talk about the ascent of sap. Now, there are few terms like what is flaccidity, what is plasmolysis, what is turgidity. You need to define these terms. You should know what these terms are. So, when you talk about flaccidity, it is when the cell wall, when the cytoplasm has completely retracted from the cell wall because of the loss of water from the cytoplasm, because it is placed in a hypertonic solution, right? So, that is your flaccidity. Turgidity is when you have complete distended cell, that is the cell is completely tight because of the influx of water, because it is placed in a hypotonic solution. Plasmolysed cell is a cell in which the cell wall has cell membrane has retracted from the cell wall. So you should know the exact definition of these terms and the conditions. Okay, the question can be identify the condition in which the cell is. What will happen to the cell if it is placed in this solution? These type of questions are asked in the examination. Is that clear? Next we talk about ascent of sap. There are four forces required for the ascent of sap. One is called the root pressure. What is root pressure? Because of continuous intake of water, there is a kind of pressure that is developed in the root cells which forces the water move to the next cell depending upon the concentration of the cell sap of the root, right? This is called the root pressure, but root pressure is not enough to make the water rise toward taller trees. Reason is gravity becomes a limiting factor, or it becomes a stopping factor for the movement of water. Then what else? Then there is something called capillary action. Capillary action means whenever there are fine tube like structures, they tend to take water. Okay, that is, they tend to make the water rise in the capillary. You must have observed when you put a straw in the cold drink or water, the level of water in the straw is slightly upper than the level of water in the glass or in the bottle. That is the capillary action. For taller trees, there is a process that is called transpirational pull. Transpirational pull leads to a kind of suction pressure which sucks in the water from the soil directly to the leaves. Okay, and the last one is cohesion and adhesion. Always remember, cohesion means the molecules of water attract to each other and forms a continuous column of water in the xylem vessel. Similarly, the xylem vessel and the water also has a kind of force of attraction which is called adhesion, which helps in the water to move in continuous manner without getting, what to say, without breaking the column of water. This is how the plant absorbs water by its root, and there is the process by which the water is absorbed is called the ascent of sap. The entire phenomenon is called ascent of sap. So water moves molecule by molecule, molecule by molecule, and finally reaches the leaves. This is all. This is what is important for the chapter. There are few more words like guttation and all, bleeding, guttation. We'll talk about it in the chapter that is transpiration. So next topic is transpiration. So this is the complete view of what is important from this chapter. The definition of transpiration is asked: loss of water from the aerial parts of the plants in form of water vapor is called transpiration. In transpiration, you will find that the leaf there is loss of water in the form of vapor, which can be detected by a paper which is called a cobalt chloride paper. The cobalt chloride paper is blue when it is dry. It is blue when it is dry, and it turns pink when it is wet. So, the moisture is detected by the paper turning its color from blue to pink. So, this is how the water vapor is tested, that the water vapor is present. Now, cobalt chloride paper, it's very important. You should know what is the function of cobalt chloride paper. When you talk about transpiration, there are three different types of transpiration. One is called stomatal transpiration, lenticular transpiration, and cuticular transpiration. What is stomata? Stomata are the minute openings present on the epidermal surface of the leaf. Please, this is the location of stomata. Stomata is present on the epidermal surface of the leaf, generally concentrated on the lower side, but it is also present on the upper side of the leaf. So, you are going to mention the location as it is located on the epidermal surface of the leaves. Okay. Now, how the stomata looks, this is how the structure of stomata is. It is the entire stomatal apparatus which includes the epidermal cells, the guard cells, the stoma. Stoma is the opening. Now when you are drawing, you will get marks only when the inner wall of the guard cell is thick, outer wall is thin, and you have the neighboring cells with the cell wall. Please mark, children, chloroplast is present in the guard cells, and this is only responsible for the initial photosynthesis and opening of the stomata. Okay, so this diagram is the complete diagram for the stomatal apparatus, and you will get marks if everything present here is there in your paper. Now, when you talk about transpiration, there are certain factors which affect the rate of transpiration. Transpiration in simple term is like evaporation. Okay, so whenever water has to evaporate, what are the general conditions? You should have a big surface area, therefore the evaporation becomes faster. You switch on the fan, the cloth dries faster. Why? Because evaporation becomes faster. When it is very humid, evaporation is low, therefore rainy season may the clothes do not dry, right? So transpiration, the factors are same. If you the rate of transpiration increases if you have more sunlight, that is heat, there is more wind, that is the wind is flowing with certain velocity, there is low humidity in the air, there is low atmospheric pressure. Now rate of evaporation, rate of transpiration increases with the decrease in the atmospheric pressure, and rate of transpiration increases with the increase in carbon dioxide concentration till the concentration reaches to around 0.03%. After that the stomata closes and hence the transpiration also goes down. Right, so this is what the factors all about. Now the plants try to conserve water by certain adaptations. Those adaptations helps to prevent transpiration, because out of 100% of the water that the plant absorbs by the root, 99% is lost by transpiration. Only 1% is used by the process of photosynthesis. So in this case, certain plants, they narrow their leaves, they don't have leaves, the leaves are modified into spines like in cactus and all. The nerium plant has got sunken stomata, so the stomata is not exposed to the outer atmosphere, the rate of transpiration is low. At the same time, there are fewer stomata, there is again cuticles are thick. If you have thick cuticles, the rate of transpiration will be low in the plants. Is that clear? Then there are certain plants like in desert xerophytic plants, they do not open their stomata during the daytime. They do open the stomata at the night time, take in the carbon dioxide, store in them and use in the morning for the process of photosynthesis when the sunlight is there. So these type of special plants, they are called CAM plants. General awareness you should know. Okay, so this is how the transpiration is all about. The maximum stomata occurs, this is all about the adaptations. The maximum transpiration occurs by the stomata, that is through the stomata. Lenticles are the small openings present on the older stem, on the bark of the older stem. Older stem is important, okay. And cuticles are the wax kind of coating present on the surface of the leaf. So the water also gets evaporated from the surface of the leaf. If the cuticle is thick, transpiration will be low. If the cuticle is thin, transpiration will be high. Is that clear? Lenticular transpiration, lenticels are open throughout day and night. So that is required for respiration, diffusion of gases. So since it is open, rate of transpiration will continue. Okay, stomata generally closes at night and it is open during the daytime. Fine. So this is all about transpiration. Now, what is the significance of transpiration? Transpiration provides a cooling effect to the plant. It also helps in the ascent of sap, distribution of minerals in the plants, and at the same time adds moisture in the atmosphere, which leads to rain. In our lower classes we have studied, right, forest brings rain. How? By the process of transpiration. Okay, there are few experiments related to transpiration, so you should be able to find out that how the transpiration is happening. Like you have Ganong's photometer, which helps in finding out the amount of water lost by the leaves, by the amount of water which is taken in by the stem, cut stem. So photometer is photo, it's not photo. Photo means light. Photo is potential, water potential. So potometer, Genong's potometer is to find out the amount of water lost by the process of transpiration, which is equivalent to the amount of water that is absorbed by the plant. Then you have Garrow's potometer. Garrow's potometer finds out the rate of transpiration on both the sides of the leaf. Since the stomata is generally concentrated on the lower side, the rate of transpiration is higher in the lower side and lesser on the upper side. So if you take a leaf and put cobalt chloride paper on both the sides of the leaf, the cobalt chloride paper which is present at the lower side of the leaf, that will turn pink faster as compared to the cobalt chloride paper which is kept on the upper side of the leaf. Is that clear? So this is all about transpiration. There are two more terms, guttation and bleeding. So when you talk about guttation, guttation is loss of water, exudation of water in the form of droplets through the margins of the leaves. And there are special structures called hydrathodes. Like stomata is to transpiration, the hydrothode is to guttation. Okay, so this is also one kind of question that can be asked. Now why do guttation happens? When the rate of transpiration is low as compared to the rate of absorption of water, a hydrostatic pressure is being created in the plants, which leads to the water ooze out from the margin of the leaves in the form of droplets. Okay, they do contain uh, dissolved components. It's not pure water. Transpiration mein the water comes out, it's very pure. It's actually equivalent to distilled water, right? Because it is evaporation and then condensation of that water. But when you talk about guttation, it has got all dissolved salts and impure salts in it as it is taken up by the roots. Similarly, there is one more step called bleeding. Bleeding is same like what happens in us. You have a cut and the cell sap and the blood oozes out. That is bleeding. In plant also, if there is any kind of mechanical injury or cut, the cell sap oozes out. This is called bleeding. This is the complete chapter of transpiration. Next we move to photosynthesis. So the next topic is photosynthesis. Now photosynthesis is a very important chapter and it has got a very good weightage for your examination. The entire plant physiology is generally asked for around 20 to 25 marks. Okay, and generally contributing 5 to 6 marks per chapter which is present there. Photosynthesis have got little bit more weightage because it has little bit more content. Okay, now what is photosynthesis? The first important question that is asked is write a chemical equation, balanced chemical equation for the process of photosynthesis. Always remember children, it's not your chemistry equation that you should have the net equation. In this case, we have 12 molecules of water and 6 molecules of water is again produced in the product side. So there are 3 products for the process of photosynthesis. You have glucose, you have oxygen and you have water. So water is reactant also and water is product also. So you have to write this chemical equation. If you write this complete chemical equation, you get 2 marks. If sunlight and chlorophyll is missing, you don't get any marks. This reactant plus sunlight and chlorophyll is 1 mark. Sunlight and chlorophyll plus product is another mark. Now always remember that the light intensity or the light wavelength which is very apt for the plant for the process of photosynthesis is blue and red. Plant do not require green color. That is the reason why chlorophyll appears green or the leaves appear green, because the light is reflected back. Which wavelength the photosynthesis happens maximum is your blue and red. Okay, so now next is the organelle responsible for photosynthesis is the chloroplast. Chloroplast is a unique organelle to the plant cell which contains stack of thylakoids which is called grana, the fluid portion that is called stroma. The yellow lines which you can see that connects the two grana is called the fret. It is two membrane organelle. So this is a diagram very important for your examination. You should know that thylakoid is the site for light reaction and stroma is the site for dark reaction. Now light reaction means it is light dependent reaction. So here sunlight is important. Dark reaction means it is light independent reaction. This light reaction is also called a photochemical reaction and dark reaction is called biosynthetic reaction. In light reaction which occurs in the thylakoids, you have two major steps. What are the two steps of light reaction? Activation of chlorophyll and splitting of water molecule. So there are two steps in light reaction. One is activation of chlorophyll and the other one is splitting of water molecule. Now chlorophyll gets activated because of the sunlight and it produces electrons which goes and helps in the process of splitting of water. What is splitting of water? The technical term is photolysis. Lysis is breakdown of water in the presence of light. So photolysis of water, the equation is two molecules of water in the presence of light. This is very important. If you don't write light, it is just hydrolysis. It cannot be photolysis. For photolysis, light has to be present. So 4 hydrogen plus oxygen and 4 electrons. These electrons are responsible for reduction of NADP or formation of NADPH, okay, from NADP, which is produced in the dark reaction. Now electrons are very important. They participate in further reactions, and now whatever is the end product of light reaction becomes the reactant for the dark reaction, and whatever is produced like NADP and ADP, they are recycled in the process of light and dark reaction. Okay, glucose is a byproduct, end product, that is the starch, and oxygen is the end product, that is oxygen is released in the process of photosynthesis. So glucose is produced in the dark reaction, oxygen and water, oxygen is produced in the, so glucose is produced in the dark reaction and oxygen is produced in the light reaction. Is that clear? Now what happens, you can see here photolysis of water, the equation is very important. Definition of photolysis is very important. The end product of light reaction is NADPH, oxygen and ATP. NADPH and ATP will go into the stroma for the dark reaction to happen. Oxygen will be released. Now this process of ATP production, how is ATP produced? ADP plus inorganic phosphate will give you ATP in the presence of light. So it is called photophosphorylation. Photophosphorylation is production of ATP in the presence of light. Now these are the end product of light reaction. Right? Now dark reaction happens in the stroma where glucose is produced, NADP and ADP, which again participates in the light reaction. This glucose molecule now gets polymerized to starch. The process by which the different single units of glucose combine to form starch, a polysaccharide, is called polymerization. So this is polymerization after the dark reaction. Is that clear? Now which dark reaction. Now which part of the plant is responsible for photosynthesis? It is the leaves. What make leaves apt for photosynthesis is the structure of the leaf. The thin flattened structure which is exposed to the sun. The upper part of the leaf, generally in the dicots, you can see it is dark in color, and the lower part is lighter in color. Upper side has higher concentration of chlorophyll so that it can trap more and more sunlight. So the large surface area of the leaf, the arrangement of leaf in the plant, and the numerous stomata present on the leaves makes leaf apt for the process of photosynthesis. Now photosynthesis, leaf has got minute structures which is called stomata. We have just understood about stomata in the previous segment on transpiration. Now stomata opening and closing, there are two theories given in your syllabus. One is potassium ion theory and one is sugar concentration theory. Both the theory works on the principle of hypertonic and hypotonic solutions. Now what happens when the sunlight hits the leaf, first beam of sunlight, the guard cells has got chloroplast. So the photosynthesis will begin in chloroplast and ATP production will happen in the guard cells. Because of ATP production, there is influx of potassium ions inside the guard cells. That influx of ions in the guard cells increases the tonicity of the guard cells or increases the concentration of the guard cells, making it hypertonic as compared to the neighboring cells. So water from the neighboring cells will enter into the guard cells and the guard cell will become fully bulged out or turgid. That leads to the opening of stomata. At night, when photosynthesis is not possible and all the ATP is utilized, for the production of glucose, the concentration of ATP drops. That leads to efflux, that is, the potassium ions move out of the guard cells, decreasing the concentration of guard cells, making it hypotonic as compared to the neighboring cells. So water starts leaving the guard cells becomes flaccid, and the stomata closes. This is the opening and closing of stomata on the basis of potassium ion theory. The next one is very simple, sugar concentration theory. As photosynthesis begins in the guard cells, so glucose is synthesized. Increase in glucose concentration in the guard cells increases the sugar concentration in the guard cells, making it hypertonic, and the process is same. As it becomes hypertonic, water moves inside the guard cells bulges out, the stomata opens at night when glucose is all utilized, the concentration of guard cells drops down, so water starts moving out from the guard cells, making it flaccid, and the stomata closes. So this is how the stomatal opening and closing is all about in the stomata, and the two theory related to it. Okay. Now, what are the factors which affect the process of photosynthesis? The factors is sunlight. As the process of photosynthesis, as the intensity of sunlight increases, photosynthesis also increases up till certain level. After that, it becomes constant. Then you have carbon dioxide concentration. Increase in carbon dioxide, increase in photosynthesis up till certain level. Increase in temperature. Now, temperature, you have a different graph. Where should I show? Now, for temperature, the graph is bell shaped. That is, there is an optimum temperature where the photosynthesis is maximum. Optimum temperature for photosynthesis is 35 to 40 degrees centigrade, where the photosynthesis rate is maximum. Below that temperature, the rate goes on decreasing. Above the temperature, the rate goes decreasing. This is the graph for temperature. For all other factors, the graph goes like this, where you have initial rise with the increase in concentration, then the process becomes constant. Is that clear? Then the internal factors like the water content in the leaf, the structure of the leaf, the cytoplasm of the leaf or the protoplasm of the leaf, these are all internal factors responsible for photosynthesis. So this is all about photosynthesis. There is one important aspect from this chapter, that is the experiments. Before experiments, what is the significance of photosynthesis? It is food for all and oxygen for all. So it prepares glucose, which forms, you know that plants are the producers. In all the food chain, the first organism is a plant. So the food is being synthesized by the plant, and plant being autotrophic are the only organisms which are capable of converting the solar energy into the chemical energy, right? And it gives oxygen for all animals and humans to survive. That is the significance. Next important part of this chapter is certain experiments. You have to know that sunlight is necessary for photosynthesis. How will you prove? Take a black paper, put it on the leaf and do the starch test. Carbon dioxide, take KOH, potassium hydroxide solution or soda lime which absorbs carbon dioxide and perform the starch test, right? That is how we are going to study about the experiments based on photosynthesis. Is that clear? Another one is there are few chemicals which are used, like what is the function of sodium hydroxide or potassium hydroxide. So they help in absorption of carbon dioxide. They actually help in making an atmosphere carbon dioxide free. The next is why is the leaf boiled first? So the leaf is boiled so that the cells are killed, so no more photosynthesis occurs. Then it is also treated with methylated spirit or ethanol to remove chlorophyll. So the leaf, what you get is pale yellow, and then you can do the starch test. For starch test, we use iodine solution. The original color of iodine solution is brown. So when you treat anything which has starch, the brown iodine turns blue-black. There is no other color, blue-black color. So the part of the leaf, now if suppose I am doing it for sunlight and I covered this part of the leaf with the black paper, here there will be no photosynthesis. So except this part, the entire leaf will turn blue-black, showing that there is no photosynthesis happening here. Now one more thing you have to keep in mind, that before performing any experiment on photosynthesis, the plant is kept in dark for 48 hours so that whatever is present, whatever starch is present in the plant is utilized. That is called de-starching of the plant. So I hope you understood the entire chapter. Next, we talk about chemical coordination in plants. In chemical coordination in plants, you are going to study two separate topics, in which one is phytohormones, that is the plant hormones, and the other one is a tropic movement. Now, when you talk about the plant hormone, they are not very complex as what we have, or the humans and the animals have, the endocrine system as such. They are simple, but there are specific function of each plant hormone. In plant hormone or phytohormones, you will study about auxin, IAA, indole 3 acetic acid. This is very important for the full form. ABA, abscisic acid. Indole 3 acetic acid and abscisic acid, abbreviation questions can be asked. So, auxin, cytokinin, gibberellin, abscisic acid and ethylene. Ethylene is the only gaseous hormone present in the plant, generally responsible for ripening of the fruits. Okay, abscisic acid is a stress hormone in the plant, and it is the only growth inhibitor that is there in our syllabus. There are many other like brasinosteroids and all that, but that is not included in our portion. So abscisic acid is a plant growth inhibitor. It promotes death and senescence in the plant. Ethylene also promotes ripening of the fruit, which is closer to the death, you can say. So it promotes senescence, but still it helps in the growth, that is in a positive development of the plant from a raw fruit to a ripened fruit. Auxin, cytokinin, they help in the stem elongation, shoot growth, root growth. Okay, and gibberellin helps in the stem elongation by internodal growth. So gibberellin is generally sprayed on the plants which are dwarf to increase their height. Okay, auxin and cytokinin, the entire table will be just present in front of you. You have to be very thorough with this table. Okay, you should know unique function of each and every hormone of the plant. Okay, now there are certain terms that you must know, like for example senescence. What is senescence? It is death. Abscission is breakdown. When the leaf breaks, it is abscission. Then apical dominance. Auxin is responsible for apical dominance. The name is so simple. Apical is the shoot tip, and dominance is it will grow first. When you remove the apex of a plant or the apical bud of the plant starts becoming bushy, or the lateral growth increases, the branching increases. That is a very important step in horticulture or in gardening. Okay, in park and all, you must have seen the plants, they are very bushy, but when we plant it at home, they are not that bushy. The reason is there is a continuous pruning that happens, which continuously chops off the apical bud of the plant. So the lateral branching increases. So the plant becomes bushy. Okay, if you want the plant to grow in height, we spray gibberellin or gibberellin, whatever you say, gibberellin. Okay, now that helps in the internodal expansion, so the plant increases in the height. Ethylene is a gaseous hormone responsible for ripening. So we use artificial ripening methods in mangoes and all. You must have seen calcium carbide reacts with water to produce ethylene, and ethylene is responsible for the ripening of the fruits. That is the artificial method of production of ethylene. Even when you have a ripened fruit and you store it, or grains, in grains we generally store the raw mangoes and all. The reason is same. The grains release ethylene, which helps in the ripening of the fruit. Is that clear? So this is all about phytohormones. Please go through the table that I have shown before. Now next is tropic movement. What is tropic movement? Movement of the plant, that is growth of the plant. You should have the term directional. So directional growth of the plant towards the stimulus is the positive tropic movement. The directional growth of the plant in opposite direction to the stimulus is the negative growth of the plant, negative tropic movement. Like phototropism. Phototropism is because of auxin. So phototropism is when the shoot bend towards the sunlight. Okay. Now so the shoot of the plant shows positive phototropism. The root grows towards the gravity. So that root shows positive geotropism. But if I have to write for the shoot in terms of geotropism, shoots are negatively geotropic, roots are negatively phototropic. Thigmotropism is touch, like the tendrils or the support. If the tendrils of the leaf or of a plant, example the pea plant, touches any support, it grows around that support, right? So that is thigmotropism. Thigmo is touch. Please children remember, touch me not plant, that is Mimosa pudica, is not a tropic movement. It's a nastic movement, thigmonastic movement, that is different. So thigmotropic movement mein you have to talk about the positive growth of the plant. So when the tendrils come in contact with the support, it just goes around the support. That is thigmotropic movement. Mimosa pudica is not an example of thigmotropic movement. Chemotropic movement is with the chemical stimulus, like the pollen grains, they germinate towards the ovule, that is called a chemotropic movement. Heliotropism is generally shown by the sunflower and the species of the same plant, which tends to turn towards the sun. This is also under the influence of oxygen, okay, and this movement is called heliotropism. It is also a kind of phototropism, but the growth is not towards the sun. It just faces the sun, okay. So the movement of the plant facing the sun is a heliotropic movement. It also happens because of the auxin hormone. So this is all about the chemical coordination in plants and what is important from this chapter. There are few previous year questions which will come after this. Go through the same and see that how many questions you could answer. Right? This is day 2 with unit 2 completed. Thank you so much children. Keep studying, keep learning. In case of any doubt you have, put it in the comment section. Okay? Your query will be answered. And be prompt with your studies. We have to finish the entire syllabus in just 6 days by giving 1 hour daily to biology.